So YouTube live is started. Internet connection. Okay. Hello, sir. Hello. Hello, sir. Yeah. So I will start with the introduction, and I will close my video. Like video will not be visible my video, so that internet trouble will not be there. Like it will be low bandwidth. So for that reason, I will close my video. So your video can be on, and the PPT you can share, and you can do things. Okay, sir. So first I will give a small introduction, and then I will give it to the session, so you can continue with the session. Okay, sir. So just I will switch on the video visible to the audience, and also yeah. First of all, I would like to welcome all the participants to 25th session of AARI Bioscience Worldwide webinar. I am very much proud and happy to introduce today's special guest, Dr. Souvik Mitra, Assistant Professor and Head, PG Department of Botany, Darjeeling Government College, West Bengal, India. He has completed MSc and PhD in Botany Department from University of Calcutta, and for his PhD research he was working on lipid profile of some genera under bryophyte of eastern Himalaya, and he has published more than 20 research articles in both national and international journals, awarded with outstanding presentation in 3rd Regional Science and Technology Congress, West Bengal, organized, and also he has got various other awards like Best Oral Presentation Award in International Conference Frontier Biological and Environmental and Medicinal Sciences, and also he has received international travel grant for presenting research findings at Natural History of Museum in 2012, and he has received various projects and awards, and he has attended many conferences and presented various projects works, and he has various field of research works like lipid classes, analysis of lipid classes, fatty acids including as derived metabolites, oxyplin pathway, and other biological significance. It's his major research area. Okay, this oxyplin pathway is a major research area of him, and today he is with us for presenting his work entitled Oxyplins, Phyto-oxylipins, Eco-physiological significance and analysis methods. Okay, so Hope you this session will be good for everyone and you people will enjoy it. And sir, the floor is yours. You can start your lecture.
Uh, Thank you for this introduction. First of all, I'd like to thank the organizing committee of 25th session of AARI Bioscience Worldwide Webinar Series organized by Annakkili Amma Research Institute. So I'm presenting my screen. So today the topic of my talk is phyto-oxylipins, eco-physiological significance and analysis methods. So, as many of the students will be there as participants, so I'll start from the basics. So, what are the oxalipins? So, oxalipins are basically a diverse and complex group of metabolites that metabolites are derived from polyunsaturated fatty acids. So, these compounds are formed from the polyunsaturated fatty acids by oxidation. So this oxidation can be enzymatic or non-enzymatic. That will I'll come in detail later. But now these oxalipins are mainly generated in much higher amount due to biotic and abiotic stress responses. So these oxalipins are of various forms. They can be present as free form. They are basically the modified fatty acids. Modified means they can be hydroxy, hydroperoxy, oxo, or epoxy fatty acids. So there can be modifications in their fatty acid chain. So these compounds are also considered as oxalipins. There can be ether compounds, there can be jasmonic acid, there can be jasmonic acid derivatives. So all these compounds are there in this oxalipin, total oxalipin. So they actually include, there can be also low molecular weight volatile metabolites. So volatile metabolites means just short chain products which are of low molecular weight. There can be aldehydes, there can be ketones, there can be alcohols, there is short chain alcohols or short chain aldehydes. So these are basically the free oxalipins, which I will talk about. But this oxalipin composition this diversity may be increased by their esterification with membrane lipids. So these modified fatty acids can be esterified with the membrane lipids, and the diversity is becoming increased. So these type of these esterified phospholipids or esterified glycolipids are now discovered. So these total composition of oxalipins are huge actually, and these oxalipins can also be present as conjugated to amino acids. So these oxalipins can be, these modified fatty acids can be conjugated to amino acids or other metabolites, so that there can be conjugated oxalipins. Now, first of all, what is the significance of these compounds? So these compounds are basically stress responsive products. So due to any kind of abiotic stresses like mechanical damage, or any kind of biotic stresses like insect or herbivore attack, these stresses can produce some stress responses, and these oxalipins act as these stress responsive products. And they also act as important signaling molecules in between plants or within plants, and these will act for ecological interactions for defense purpose. So basically these compounds are acting for a defense response. They also can act as hormones, and there are some reports that oxalipins may also act as antimicrobial as well as antifungal compounds. Now, there are also reports in case of algae, these oxalipins mediate certain resistance to diatoms against algacidal bacteria. So there are huge significance of these oxalipins if we consider basically the defense purpose. So they are basically helping in defense response, and also there are some developmental purpose for which oxalipins are also play a significant role. So what I was talking, that oxalipins may also be free form, may be present in free form, or they may be present as esterified with some membrane lipids. So free form of oxalipins, or we will basically focus on the free form of oxalipins, because this total diversity is huge. This total diversity of compounds are huge. So you will see that the free form of oxalipins are also huge classes of these compounds you can see. So these free out of these free forms, there can be some derivatives of fatty acids. Derivatives means you know that fatty acid structure, basic structure is that there is a carboxyl group and there is a hydrocarbon chain. So any kind of modification in this hydrocarbon chain due to oxidation, that can form this kind of modified fatty acids. So these all will act as the oxylipins. Now you can see this hydroxy fatty acids means in this hydrocarbon chain at some point there is a hydroxy linkage, so that will form a hydroxy fatty acid. There can be keto group in a fatty acid, that can form keto fatty acids. There can be hydroperoxy fatty acid, that is hydroperoxygenation at this fatty acid chain. Hydroxy epoxy fatty acids, so epoxy and hydroxy both are there. Similarly epoxy fatty acids and divinyl ether, so they are basically the modified forms of fatty acids. So these can be one type. Another one, free oxylipins, are the green leaf volatiles. So green leaf volatiles are also free form of oxylipins. So first, these modified fatty acids that I was talking in the last slide, these modified fatty acids may also they can also cleave to form short chain compounds. These modified fatty acids can cleave to form short chain compounds. So these short chain compounds means they can be 6 carbon, 5 carbon compounds, there can be 8 carbon compounds, there can be 9 carbon compounds. So these short chain compounds can be formed. So you can see that here I have given the structures of some volatile, these low molecular volatile compounds. These will also be considered as oxylipins. So here I have given some 6 carbon compounds. The examples of some 6 carbon compounds you can see, there can be 6 carbon aldehydes. Obviously 5 carbon, 7 carbon, 8 carbon, 9 carbon aldehydes can also be there. So 6 carbon aldehydes, there can be saturated aldehydes, that means without any kind of double bond in their hydrocarbon chain. There can be unsaturated aldehydes. Okay, so there can be saturated aldehydes, there can be unsaturated aldehydes, there can be saturated alcohols, there can be unsaturated alcohols. So this total composition is termed as the green leaf volatiles. So main leaf volatiles are basically 2 hexenal, which is termed as leaf volatiles, leaf aldehyde, and 3 hexenol, which is leaf alcohol. So basically why they are called as green leaf volatiles, because if you mechanically damage a leaf, you will get some smell. That smell is basically due to these leaf volatiles. So these leaf volatiles are emitted and you will get that smell. So that is why these are termed as green leaf volatiles. So green leaf volatiles are one group of oxylipins, and they are obviously they are free form and they are of low molecular weight. So these all are free oxylipins. So modified fatty acids and their cleavage products all will be together considered as free oxylipins. Next, esterified oxylipins. What I have told that this structural, this very diversity of oxylipins is high, and it has now become very high due to the discovery of different kinds of esterified oxylipins. So if we consider that in membrane you can get phospholipids and you can get also glycolipids, that phospholipids and glycolipids, you know that there can be fatty acids attached to the glycerol moiety, and these fatty acids, if there is some modification due to oxidation of these fatty acids, that can form these modified fatty acids. So these modified fatty acids can get attached with these phospholipid molecules, so this will be modified phospholipid. So these are oxylipin esterified with phospholipids. So if these phospholipids can be esterified with modified fatty acids. Similarly, in case of glycolipids, this can also be esterified with modified fatty acids. So one example I want to specifically mention, the last one, that arabidopside C. In this case, the digalactosyl digacyl glycerol, this DGDG, this glycolipid is modified. How it is modified, the fatty acid chains, the two fatty acids which are attached with this glycerol, these two fatty acids are basically modified fatty acids, which are name is oxo 12 oxophytodienoic acid. So these are some modified fatty acids